and we were beginning to talk about how the two helical halves hold together, and the bonding between the base pairs is very significant, and it allows us to think about ways in which genetic information is transferred, either by copying itself and or by using the DNA template to create other entities within the biological system that are functional. And so it's important, and we all know that G's and C's form 3 hydrogen bonds. All right, but this is what class? Biochemistry. So we're learning the chemistry. So this is an important slide to actually know how the structures form those three. And so the structures of the guanine, cytosine, and how they form the three bond pairs. Very, very important. AT form 2 bond pairs, hydrogen bond pairs, and knowing how those structures create those bonding pairs. And so you know a hydrogen bond occurs between X, which could be nitrogen, oxygen, and fluorine that has to be covalently linked to a hydrogen. And then on the other end, X could be oxygen, nitrogen, or fluorine has a lone pair. The hydrogen bonds forms between them. And so we have a bonding pair for 2 for A's and T's, 3 for C's and G's. That's very, very important. All right. Something else about DNA size. Do you see this little dot right up here? Right there? E. coli. That's a cell. Here is E. coli's DNA unraveled. There seems to be a disparity there, right? That long strand of DNA. Now, here's the other part of this. When you take a look at that, the DNA is not that thick. Okay, we're showing in terms of relative size, so we can actually show it to you. It would be a very, very thin line, almost one that you couldn't see on this scale, but the length of it is proportionate. There has to be some way that DNA gets inside that E. coli cell, because it doesn't even take up the entire volume of the cell. Okay, here is a representation in which the DNA is linear, or all the way as linear as it can be. DNA for E. coli cannot be linear. Why? Why can it be absolutely linear? It's a circular piece of DNA, okay? But there's no additional structure involved, okay? In a circular piece of DNA, we do see that DNA compacts, and the way in which it compacts, it supercoils. All right, and so it twists around itself, and you can see that it's larger when it's fully unwound, and as it winds up, it gets into smaller and smaller entities. It's kind of like if you've ever had a necklace and a chain, and you let it in the bottom of your jewelry box, it gets all knotted up. It becomes more compact, but it becomes complex. Now, what's the difference between like a string or a chain that gets all knotted up is that many times this is absolutely controlled, in which it can be unraveled and raveled back together in an ordered way. Although you get a necklace and a knot, sometimes just can't get undone. There are 2 ways in which we measure the compact, or the twisting and the writhing, that we call it twist and writhe. These are both elements of supercoil. And so what we're doing is we're just compacting it by twisting it in some fashion. There is negative twists and positive twists. I don't care that you can calculate this. I just want you to understand that there are different ways in which we can do this. The reason why I don't really care about the circular bacterial is because in humans, what we do is we compact our DNA using proteins to facilitate this. And so DNA has a charge associated with it. What charge is that? Very, very negative. we take proteins that are very, very positive on the outside, and we wrap the DNA around the protein. So here is the DNA wrapped around the protein. The protein is here. We can see the protein in a crystal or a space-filling model. The DNA in the that is blue or purple, I can't quite tell, wrapping around it. And so it is organized. And so what we do is we make this wrap around. Now, the problem is to make it wrap around, sometimes we have to reduce the strain as we coil it up. And what we do is we use a protein called topoisomerases, which can cut the DNA specifically, and then ligase or recombine it back so that as we twist it up, we don't create additional strain. But we do create additional order by wrapping the DNA around the histones. The histone proteins are not a single protein, but a complex of proteins put together. And they are relatively small in molecular weight compared to proteins. And they are very high in 2 amino acids. Lysine and arginine, we'll learn lysine and arginine in the next chapter, are positively charged. And so in terms of percentage, some of them can be as high as 30% positively charged on the outside. This makes a wonderful reason why DNA would want to bind to this particular protein. And it is very ordered as it does that. The topoisomerases, there are 2 types, type 1 and type 2. Okay, what is the difference? It's the way that they cut and relieve strain. Type 1 will cut 1 strand of the DNA, and so the DNA kind of unravels only by 1 strand, and it can unravel and then be ligased or connected back together, creating a little bit of openness, less strain. Topoisomerase 2 cuts both strands and can actually move the DNA much more rapidly to release strain. The topoisomerase 2 activity, not only does it cut both of them, but it has to hold on to both strands to bring them back together. So there's a little bit more coordination in that. Now, DNA is very, very complex in its structure within the mammalian system. Here's an example. We showed you before that DNA double helix wrapped around a histone. Let's show you a little bit more about that organization.
Of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are divided. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay. Why would the less highly organized structure of DNA be important? Why isn't it always compacted like this? What is that? Yeah, what you have to do to use the DNA to produce the things that we need to get down to a single strand of it in a single linear sequence. Not only that, we also have to split it apart into its two strands so that we can use it. And so if it's all tightly wound all the time, we would never be able to get to the genetic information. But if it was totally unwound all the time, it would take up way too much space. And so there is a balance between what is accessible, what is not accessible, and how it's compacted. That is important for the control mechanisms and to be able to produce the types of things that we need to produce and the sections of the DNA that we need to access. This is even different between different tissue types. Okay? Because there are certain proteins that are upregulated in certain tissue types where those proteins are really not upregulated in others. And so the control mechanisms about how we access the DNA is not generalizable to all cells, but are very unique to different cell types. Now, in general, if we take a double-stranded DNA, we can unravel it. Okay, another name that we can call is denature it. Okay, pull it apart, and we can. Another term that we talk about also is we melt it, and we can get two single-stranded copies of the DNA. Okay, if we allow it to cool, or another name for that is to anneal back together, they actually come back and form the double-stranded exactly as they were when we first started. Okay, and so we can denature it or melt it, and it can anneal back into a perfect alignment and come back. The reason why it comes back in perfect alignment is the base pairing and the sequential order of how they come together. Now, in this process, okay, how in the world can we go from the top to the bottom? What can we do to actually cause it to separate? Well, melting gives you an idea. We can add what to it? Temperature. Okay. So in a laboratory, we can put DNA in a test tube, we can heat it, and we can separate it. This technique using heat to separate DNA is used all the time. Okay. You may have heard a process called PCR, in which you have to, the whole point of the instrument is to this: heat it up, make it hot, cool it down, let it sit for a certain temperature, heat it up, cool it down, let it sit at a certain temperature. That's the whole instrument. From that, we can do very powerful things because the DNA will open up, it will come back, it will then be heated up to do a reaction, and then we'll start the process. Open it up, allow it to come back together, do a reaction. And so that's important. We can also add acid or base to denature. Okay? How in the world does acid and base denature DNA? Obviously, heat, you give it enough energy, you're going to break all bonds, right? But how does acid or base?
Changes the charge. Subunits.
It could change the charge, okay? In this case, it could take away, we can protonate the negative charge and make it neutral, okay? But we're not held together as much by charge. What is holding most of the strands together in a linear sequence? The what? The hydrogen bond. If you put acid in, what can you do to the hydrogens? You deprotonate them. You take off all the protons, you get a hydrogen bond. And if, conversely, if you put acid in this situation, the nitrogens can be protonated. Okay? Or those other entities that have lone pairs that make the hydrogen bond, they can be protonated. And so you remove the hydrogen bonds by adding acid or base. Under certain conditions, we do use temperature. Under certain conditions, we usually use base, mild alkali. You can use acid. We don't usually use acid to denature DNA structures. The integrity of the DNA strand itself has a certain level of integrity. And when you do experiments, you either increase the temperature or you increase the concentration of the alkali, and you will get unraveling as you go through that. What really happens, though, is that the unraveling is pretty quick. Okay? Now, what affects melting temperatures? Okay, there are a lot of sources of interaction. Most of the time, we talk about the hydrogen bonds. We think that is fundamental, but it's not the only interaction that holds the DNA together. There is also hydrophobic interactions. Those base pairs are hydrophobic, and they interact themselves. Now, if you compare a hydrogen bond and a hydrophobic interaction, hydrogen bond's a lot more energetically favorable, but you can't dismiss the hydrophobic interactions. Also, the DNA pairs themselves are aromatic. So they have pi bonds that are conjugated, and as the DNA, we know the hydrogen bond this way, but they stack on top of each other. That pi-pi interaction above and below causes stability. So we would have to overcome that. Also, the electrostatic interaction. We do have a lot of phosphates, okay? But what we also having roaming around the system are lots and lots of magnesium. Remember, even in the standard energy, free energy, we said it has to be 25 degrees, pH 7, 1 molar concentration, and they also give you a concentration of magnesium. This is why magnesium's in the mix. Magnesium is bridging these the phosphodiester bonds, and that actually creates electrostatic interactions or salt bridges that hold the structure together. If we unravel the DNA, we have to displace that phosphate magnesium bond that holds it in its structure, and so there are multiple energies that hold the DNA structure together. When we start to melt or unravel or denature the DNA, it usually happens over a very well-defined period of time or concentration of alkali or temperature. Okay, what happens is this: the bonding is cooperative. Okay, it is something like this. Have you ever tried to put on a pair of jeans that were just a little tight? Okay, so you're trying to get everything lined up and it's not lined up. You lay down on the bed and you try to pull on it, but as soon as you get everything lined up, once you get the first one going, it all goes, right? Zips up. Then you can't breathe. Okay, but you're looking good. All right. And the same thing happens when you're trying to unzip it. Sometimes you just get it going, but as soon as everything lines up, as soon as you let the one go, that helps the next one go, that helps the next one go, and then you can breathe and everything's better, right? And so these cooperative, once you start breaking them, they all tend to break, and once you start making them, they all tend to make. You can see that this way in terms of the melting curves. Lots of integrity, okay? In terms of here, you've got temperature. Nothing is breaking apart. But as soon as you get to a certain temperature, it doesn't take long, maybe 5 degrees over a period for this one, before it totally denatures. This one's even tighter. Over maybe 1 or 2 degrees, it goes from almost full integrity to fully denatured, fully unfolded. And so the measure that we take is always at the 50% mark. And so 50% folded is a melting temperature, and we can compare the stabilities of DNA at the 50% mark. Now, what you can probably deduce is that the higher concentrations of GCs, you can see what happens to its melting temperature. The melting temperature goes up linearly in a line as you increase the concentration of G's and C's. And so you can actually get DNA that has integrity even at 100 degrees Celsius or boiling water. That's important. because there are entities that live in environments that are very harsh. There are quite a number of entities that live near the bottom of the ocean where you think it's really, really cold, but where volcanoes are coming up. And that water temperature is above boiling, and there are creatures roaming around in there, living, being happy. All right? You put us in 100-degree water, we're not happy. Okay? Kind of interesting. We actually have isolated certain proteins from those creatures that we now use in laboratories because they can withstand very high temperatures, and we just kind of hijacked their ability to be available and still active in very harsh conditions. The genetic information, genes of all eukaryotes and prokaryote organisms are made of DNA. Viruses, genes, are made up of either DNA and or RNA, depending upon what you find. The genetic information is transferred by RNA and R systems. And if you take a look at RNA, RNA looks exactly like DNA in terms of its structure. The only difference is this 2' hydroxide. Now, this 2' hydroxide has a consequence. All right. This 2' hydroxide makes RNA liable, or it degrades really quickly. Why does the 2' hydroxide have an effect on the stability of the information? Where is the information in RNA and DNA? Where does the information reside? In the what?
The bases.
In the bases. Not just the bases, the sequence of the bases, the connectivity. As soon as you break the chain, you've broken the information. So why is this 2' hydroxide make RNA less stable? Yeah, the bonding or its reactivity. What can happen is base can take that proton. I now have a negative charge on that oxygen. Okay, that negative charge can attack, kick up, kick down, and break the bond. Okay, it has that inherent ability. It can do it by itself. Although there are lots of enzymes that are available to facilitate this, to break RNA down. It can do it by itself, but there are enzymes to do that. Once you've broken the chain, you've broken the linear information. Without that hydroxide, the stability of the linear sequence is much, much better. And so DNA is a much better genetic carrier in terms of stability, structural stability, but it has been theorized that RNA was the original genetic information transfer molecule even before DNA was developed. RNA has a host of unique abilities. One, it can hold genetic information. Two, RNA can actually facilitate reactions. It can be a catalyst. It actually can be an enzyme. So it can have genetic information and it can be an enzyme. If you look also at all the fundamental transfer molecules in the body, including energy, electron carriers, cofactors, all of them seem to have RNA as part of the structure, and the derivatives of the RNA create the function. With essentially every bit of the biological, all the processes that we do can fundamentally come back to RNA. There is an idea that RNA is the original functional protein, or the functional biological entity, before proteins and before DNA. Genetic flow, again, we've seen this. DNA to RNA to protein is what we think about, because proteins, for a lot of biochemists, were the ultimate important piece. Okay? And so the gene is a strand or sequence on the DNA, and the gene product, for a lot of times when you talk about this, is a protein down here. But now we know gene product can be a whole host of things. Any entity that has biological significance that is derived from the DNA, okay, is essentially the outcome of a gene. And so the messenger RNA does transfer that information to create a protein, which is definitely a gene product, but we create all sorts of things from the DNA that are functional, like transfer RNA. We can't function unless we have transfer RNA. RNA is a transient, actually it has some three-dimensional structure. It stays around for a little bit. It is used. It is on the DNA, so it is a gene product. RNA itself does not have the ability to follow Chargaff's rules. There is no base pairing that is dictated, and so the percentages of A's, G's, C's, and U's can be all over the place. It is typically single-stranded, although we do see that it can form complex three-dimensional structures and include some double helical nature. Okay, it doesn't have to be double helical, but it can. It also has imperfect double helical structure, and so we have bulges and loops and hairpin turns as part of the structure of some RNAs. We kind of think RNA is typically one little strand and it's a single strand that goes around. That's kind of what messenger RNA is. It's one single strand. We're going to read down that strand and we're going to translate. But every other piece of RNA has three-dimensional structure, even enough to crystallize it. It's regular and repeating. RNA can function, and messenger RNA can function in several different ways. It is an informational transfer. It is at least one messenger RNA is produced for every gene product that is formed for proteins, but it can actually, depending upon how it's processed, create one messenger RNA to create one protein, or we can make one long piece of RNA, and from that RNA we can create several proteins, depending upon how it's processed. And so we can, messenger RNA is a little bit more complex in its structure. Transfer RNAs. Here is a piece of RNA that does have function. Transfer RNA will translate the messenger RNA, bring in a specific amino acid, and it will bring it into continued synthesis of protein. But if you take a look at it, that structure that is bound, okay, is very similar in all, 63 transfer RNAs that are available. Even with different sequences, they all have almost identical shape. There is the portion down at the bottom that will read the messenger RNA and make a complement to it. And then way over at the top is where we were going to attach an amino acid to it. But even though the sequences are different, the structures are the same. We have quite a bit of double helical structure and complex three-dimensional structure, even within the double helical. Ribosomal RNA is the most abundant RNA in the biological cells by volume and mass. It plays both a catalytic, ribozyme, and a structural component as part of protein synthesis. It is itself an enzyme, but it works with a whole host of proteins that are a little bit more efficient in their enzymatic activity, but the ribozyme activity is essential in the process. Small nuclear RNAs are small pieces of RNA that are present in eukaryotic cells, and they help in processing the splicing of RNA exons. And so sometimes we create much, much more RNA than we need, and we have to clip out pieces. These small functional pieces of RNA help and are enzymatic in helping cleave those pieces out. So there are a lot of different products that we can have that come from RNAs. All right. Shouldn't have been a surprise. We're going to go now and we're going to explain some experimental methods that we use with DNA and RNA. And so, just a kind of a definitional thing. Cloning. Okay? Cloning has actually changed its definition over time. It's actually become a lot more generalized. We think about clones, you make a copy of something. We have cloned things through horticulture for thousands of years. Okay? We have cloned, made copies of things that way. But now in the lab, cloning had the idea of this. You separate cells down till you get a single cell. Okay? And you isolate that one cell, and that cell will replicate. Once you've isolated one cell and you've allowed that to replicate, then all of those cells came from the progenitor, and so all of those cells are identical. You have cloned a cell. Okay? That's an idea. Everything is identical. Now, when we talk about cloning, most of the time what we're going to do is we're going to talk about taking out a piece or a segment of DNA. Okay? And take that out. We're going to insert into something, okay, into some sort of carrier, and then we're going to replicate that, either replicate the actual DNA itself, make copies of the DNA, and or we are going to make that DNA produce a protein, and so we're going to clone the protein itself. And so, to go over that concept, here is the generalized DNA cloning. All right, so way at the top, we get a cloning vector, a plasmid. How do you get that? How do you get a cloning plasmid, the vector itself? Have you ever used one? Did you do genetics? Have you done this experiment in genetics? Okay. You actually buy that. Okay. It's one that was naturally naturally found in bacteria. Okay. We isolated it. Somebody isolated it, and we sell these vectors. And there are a couple that are very widely and popularly used. Okay, and so you'll actually see abbreviations for them that are used. We chose them because how effective they are in infecting the cells that we use. A lot of times we use E. coli. You take your source DNA and you're going to cut up those DNA into fragments that are a size that are workable. Okay. We use something called a restriction enzyme or a restriction endonuclease. What in the world is that? Do you know what a restriction endonuclease is? It's going to cut DNA. Okay? Does that seem like a protein that is good for you? A protein that cuts DNA? No. Restriction enzymes themselves are naturally occurring proteins that are essentially the bacterial's immune response. Okay? So bacteria create these proteins and they cut DNA. And what it's intended to do naturally is if a bacteria gets a piece of DNA that is foreign inside of it, it's going to cut it so that it doesn't overtake its own function. Well, that's a problem. Doesn't bacteria have its own DNA? Why does the restriction enzyme not cut the bacterial's own DNA? Because it's only looking for a little tiny bit of DNA. Randomly, that's going to happen in your sequence. Do you know why the restriction enzyme doesn't cut its own DNA? the bacteria takes its own DNA and it has other proteins inside of it that methylate the base pairs to make it the wrong shape. So every bacteria has its own little restriction enzyme. It doesn't cut its own, but it'll cut anybody else's. Again, what we've done is we've isolated those restriction enzymes away from the source, put them in a bottle, and sell them to you. And so then we use it as a molecular tool. All right? And so we're going to cut some foreign DNA, and we're going to cut the vector itself. Now we cut them with the same restriction enzyme because the ends will be a perfect match. Okay, that's cool. And then we mix it up. And we allow the little chunks of DNA that we want to try to get to and the vector to mix together. Hopefully, the little chunk of DNA and the little vector come together and make a nice bond because of the overlapping DNA sequences. How often does your little chunk of DNA get into a vector? Is it a lot? The video showed every single one of them picked it up. Does that happen in real life, every one of them? No. Your insertions can be 1%, maybe 3 or 4%. Okay? So you overcome this by throwing in lots and lots of vector and lots of DNA, and you get a percentage of random confirmation. Now, the vector gets cut, opens up, okay? And when you send in the ligase, the enzyme that repairs cuts, okay, the plasmid could connect to a piece of DNA that you want. That's cool. Or it could come back and plug itself. Statistically, if it opens up, most likely it's just going to close right back up and be inserted, right? Because these two ends are already connected, so they're probably going to hit right back together. So you're going to get a lot of vectors that have just been reclosed back up, a few of them that have the target that you want. All right? We have, and this says we have recombinant vectors. Great. All right. We've got 3% of them that are recombinant. 97% of them are a plasmid itself. Okay? And then what we do is we mix them with cells. Now here's the problem. That vector in that cell, there's a cell membrane. It's hard to get that through. Picture looks like it's easy. So what we do is we have to treat these cells to make them permeable. It means we have to break apart the cell membrane a little bit to allow stuff to get in. We do that two ways. One, we treat it with calcium chloride, a certain temperature and a certain concentration that we're trying to balance, killing them versus allowing them to be leaky and get stuff in. Again, we get a few percentage. That's okay. All right. Or we shock them. Electroporation. We actually hit them with electricity. That doesn't seem to be really good either, but again, we kill some, we get some in. Now here's the good thing about it. We have a few cells. We give them time, temperature, and food. What's going to happen to the cells? They'll replicate. So we don't need a lot. We just let them grow. And that's cool. All right? Now, I got lots of things that are growing. Do I really have the DNA in there or not? Well, we allow it to grow, but we allow it to grow on a medium that has antibiotics. All right? Bacteria doesn't like antibiotics. And so they'll grow if and only if it has a plasmid inside. Okay? So most of the cells die because the antibiotic kills them. Only a few live because only a few got a plasmid inside of them. But just having a plasmid inside of them, does that mean it has the DNA that you want? No, you've got cells growing on a plate. Okay? You then have to, you then have to isolate and test to see your particular DNA. All right? A good way to do that is to hit your cells with a little bit of, or actually do this. You take the cells and you take a piece of paper. We talk about nitrocellulose paper. What we do is we take that little round plate that you're growing, we put a piece of paper on there. Piece of paper adheres some cells. And you take it off, and now you have a paper copy of what was on the plate. And here's the problem. Round plate, round piece of paper. Unless you make a mark to know where this is it, where you lined it up, you're going to have an issue. So you got to mark it. This is kind of a laboratory technique. Okay, now I got a piece of paper with lots of cells on it. How in the world do I find this out? Where things are? All right, remember that whole thing about denaturing, unraveling it? Okay, we can use that same technique to test this. But what you have to do is you have to treat the paper. Now the nitrocellulose is good because it adheres things. So a lot of times we treat it with alkali. Breaks the cell membranes and it denatures the RNA, the RNA DNA in there. So it's denatured the DNA. And so now I have a copy where the cells are. They've been all opened up. All of the DNA is open. What we do is we take a little target piece of RNA or DNA. We synthesize it because we know what we're looking for. We call this a probe. And we either attach something that's fluorescent or radioactive, and we put it on the piece of paper in solution, and where our target is, what we're looking for, will allow it to bind and adhere and anneal. If the DNA isn't there, it's not going to anneal. We wash it away, and anything that is stuck will show you exactly where that particular cell has your targeted interest. All the other cells, not your target of interest. But that's okay. You scrape off that little bit of cells, you put that in low growth medium, and now everything that grows is exactly what you want. And so you don't need 100%. Just need a little bit of time, a little bit of food, let it grow, and you've got everything that you want.